Hello, good evening to our audience and guests from Malaysia, and a very good morning to our audience and guests from the United Kingdom. Welcome to our webinar on Adiposity and Disease Risk, a global insight. This webinar is jointly organized by UKM Medical Molecular Biology Institute and the University of Oxford. We are very fortunate today to have a distinguished lineup of speakers from both institutions to share their view and findings from two sides of the globe. Over the next 2 hours, we will listen to the global perspective on adiposity, prevalence of obesity, how genomics could provide some information, the differences and linkages surrounding obesity, the approaches towards combating this universal problem, the valuable resources for this type of study, and lastly, the UK-Malaysia Health Research Partnership to address this global issue. To the audience, you may post your question online at the Q&A box, and we will highlight it to the respective speaker to be answered online. Throughout this webinar, we will allow 1 or 2 short questions immediately after the talk by each speaker. We will also have a final Q&A session after the last speaker for the remaining questions to be answered. Without further ado, let's welcome our first speaker today. Professor Sarah Lewington is a professor of epidemiology and medical statistics at the University of Oxford in the Nuffield Department of Population Health. She is also the Director of Graduate Studies and the leader of the Global Population Studies Group. Today, Professor Sarah will enlighten us with her topic on adiposity and cardiovascular disease, a global perspective. Please welcome Professor Sarah.
Thank you very much, Siok Hong. So thank you very much for the introduction, and thank you very much for the opportunity to open this exciting programme. So as Siok Fong said, I'm going to be talking about adiposity and vascular disease with a global perspective on that. So just to put that in some context, in 2019, there were 56 million deaths around the world, of which 28 of those were before the age of 70. In other words, 28 million premature deaths, of which 20 of those were in middle age, which is the age that we're largely studying in the UK Biobank and in the Malaysia cohort study. Of those 18 million, a third were due to cardiovascular disease, a further 5 million to cancer, 1 million to diabetes, and 3 million to other non-communicable diseases. So 15 million deaths in total in middle age due to non-communicable diseases. And we know from work done mainly in high-income countries that the major modifiable causes of vascular mortality are blood pressure and blood lipids, adiposity and diabetes, and tobacco. And together, these risk factors, these causes are responsible for the vast majority of those vascular diseases. And so today I'm just going to spend most of the time talking about adiposity. What do we know about adiposity from other parts of the world, not just the high income countries? What have we learned? But first of all, I just want to put that in perspective to show some results on blood pressure from around the world. So here are the results from some studies that I've been involved in Mexico, Cuba, Chennai in South India and Russia. And you see that in all of those countries, there's a really consistent association between systolic blood pressure and coronary heart disease, mostly heart attack. So here we have risk on the vertical axis and on a doubling scale, and along the bottom, the long-term usual systolic blood pressure. And you see that in all of these studies, we saw an approximate doubling in risk for every 20 millimeters of mercury higher systolic blood pressure. So remarkably consistent between these studies done in very different diverse populations. So what do we know about adiposity? And I'm going to focus mainly on BMI through the talk, but this was work that was done led by colleagues in Cambridge and Harvard, but in collaboration. And this was the global BMI collaboration. It was a global collaboration of over 200 studies with 10 million adults. But when they were looking at BMI, it became increasingly obvious that we need to avoid reverse causality. Long before people die, they start to lose weight. So in order to prevent this so-called J-shaped association, we need to avoid reverse causality. So we need to exclude people with prior disease and early follow-up. And we also, there's massive confounding with smoking. And so the analyses were restricted to never smokers. But that still left 55,000 deaths, coronary heart disease deaths among 4 million adults. And what they saw quite clearly then was at least down to a BMI of about 20. And certainly for anyone who was overweight, for every extra 10 units of BMI, there was an approximate doubling in risk. So how does this compare with those other studies that we've looked at in the low and middle income countries? Well, here's a study in Mexico City, prospective study, 150,000 adults recruited around the turn of the century. Questionnaire, physical measurements, a blood sample was taken, and linkage to national mortality database. And here, again, after very careful analysis of the data here in Mexico, where diabetes is so incredibly prevalent and quite poorly controlled, meaning that before people die, they've tended to lose weight. So excluding people with diabetes and early follow-up, again, they see that for every 10 units of BMI, there's approximate doubling, maybe slightly weaker, but an approximate doubling in risk, as we'd seen in high-income countries. But if we move to South India or South Asia, then in the Chennai prospective study, this was 500,000 adults followed up so far to 2015, but follow-up continues. Deaths found by visiting all households and linking to the death registry with verbal autopsy of causes of death. Again, to limit reverse causality, the analyses excluded people with prior disease and deaths in the first 2 years of follow-up and focused on never smokers. So this is a study run by the PI, Professor Dr. Gajalakshmi. So looking at BMI versus coronary heart disease mortality in Chennai, what did we see there? Well, we saw an amazingly different association there, with really little or no association throughout this range. And don't forget, we've excluded people with prior disease. We've excluded the never smokers. So this is among 8,000 non-smoker cardiac deaths. In this population with a mean BMI of just 23 units, we saw little or no association with BMI. And to say that this surprised us would be an understatement. And so we looked carefully at the associations of BMI with blood pressure and with diabetes. And as you would expect, we saw an association between BMI and systolic blood pressure at baseline, and between BMI and diabetes at baseline. And then when we related those to subsequent mortality, again, 20 millimeters of mercury systolic blood pressure and self-reported diabetes at baseline, again, approximately double cardiac mortality, as we'd seen in high-income countries. So it makes this flat association even more surprising under the circumstances. And so just to put that in perspective, what that means is that at any given level of systolic blood pressure, there's actually an inverse association in this South Asian population between BMI and cardiac mortality. So that's among people in Chennai, in Tamil Nadu. What's involved in different exposure and disease patterns. So we more tropical and the very industrial east of China, the rural areas. So you see urban, 5 rural areas, really quite a population within, or sample within within China. This was going to show some results from a PhD student, Andrew Iona, who looked, oh, sorry, I'm not moving on. So who looked at BMI and stroke, incident stroke, again, in middle age in China, mean BMI of 24. For ischemic stroke, again, there was a strong association between BMI and systolic blood pressure in this study, as you would expect from studies in high-income countries. And given the effect on BMI, the 10 unit on systolic blood pressure, 10 units of BMI was associated again with an approximate doubling in risk, as you expected from the effect of BMI on blood pressure. But remember, blood pressure is also strongly associated with hemorrhagic stroke. So what did we see for hemorrhagic stroke? We would have expected an even stronger association between BMI and hemorrhagic stroke based on its BMI's association with blood pressure. But again, surprisingly, instead of that even stronger association, we actually saw very little association between BMI and hemorrhagic stroke, with just a relative risk, a rate ratio of 1.2 for 10 units, whereas we would have expected a rate ratio of about 3, given the BMI effect on systolic blood pressure. So again, at any given level of blood pressure, this suggests an inverse association between BMI and hemorrhagic stroke in the China Kadoorie Biobank study. If we look, maybe it's that BMI isn't measuring body fat, but in the China Kadoorie Biobank study, they had measurements of percent body fat. And again, what we're seeing for ischemic stroke is more or less what you would have expected, 20% higher body fat associated with a relative risk of 1.6. But again, little or only a weak association at very high levels of body fat for hemorrhagic stroke. So something very different going on with body fat between ischemic stroke and hemorrhagic stroke in this Chinese population. So to conclude, what does this tell us? Well, I think it tells us that we need these big long-term prospective studies in many populations, and that we certainly can't extrapolate from the big studies in the high-income countries to the low and middle-income countries, particularly for adiposity, that we need to go beyond BMI if we're going to understand fat distribution in different populations. We need reliable country-specific evidence on adiposity and cause-specific cardiovascular mortality, those striking differences between the associations with ischemic and hemorrhagic stroke, for example, and that we need to monitor the different and evolving effects of adiposity in many populations, particularly in the low and middle income countries where the demographic is changing so rapidly. Thank you. I'm open to questions. Thank you.
Thank you, Prof. Sarah. Okay, there seems to be a question here for you, Prof. Sarah. What is your opinion on some hypothesis that fat-free mass is a stronger determinant of stroke volume and cardiac output than adipose mass or other clinical characteristics? Is there any association between fat-free mass and cardiovascular disease?
Well, I think that's a great question, and it leads nicely into the study that we're doing that Prof. Asian will talk about at the end. So we really don't understand fat distribution. I don't think the studies have been big enough so far, but we have lots of large studies coming online now that are large and have well phenotyped adiposity. So we need to look, we have the 500,000 in China, the 500,000 in the UK. We're going to have detailed DEXA scans from the study in Malaysia. And I think it's only really when we look at these things in large studies that we'll really understand what's going on. We need well phenotyped baseline, but we also need well phenotyped outcomes to look at that, because as you saw, really different associations between ischemic stroke and hemorrhagic stroke, for example.
Mm-hmm. All right. Thank you, Professor Sarah. We can
Thank
only
you.
allow one questions for now. So we will be back for more at the final session. So I would like to
Thank you.
welcome our next speaker from Malaysia. All right. So Professor Dr. Nolaila Mustafa is a consultant endocrinologist and the head of Department of Medicine in the Faculty of Medicine, UKM. Professor Nolaila will bring to us the prevalence of obesity in Malaysia. Please welcome Professor Laila.
Okay, right. Thank you, Shok Fong, for the introduction, and thank you to the organizing committee and also to our colleague in the UK. I'm no longer the head of department, Shok Fong, but anyway, so I was given a privilege to talk about prevalence of obesity in Malaysia. And we know very well it's on the rise, but just let me talk a bit about the obesity. When people talk about obesity, we always talk about a BMI. And when we talk to layperson, what they understand is, they can see that there's fat around the tummy region, there's fat around the buttock, thigh, and so on, especially for women. So basically, it's due to excess of body fat, more than 20% in young men and more than 30% in young women. And, in the scientific term, is known as adipocytes. Of course, we have, different percentage of body fat that it is acceptable for men and women. And when, somebody has excess of body fat, we call them obese or obesity. But in many studies, as what mentioned by Professor Sarah, a lot of studies looking at BMI, it is actually an indirect measurement of obesity, whereby, we measure the height, weight, and then it will correlate with the amount of body fat in an individual. So it's very simple measurement that we can correlate with the amount of fat. So in Malaysia, we have been adopting the Asian BMI cutoff point using BMI to classify obesity, whereby less than 18.5 kilogram per meter squared is classified as underweight, normal between 18.5 to 22.9, and anything beyond 23 is actually overweight. And we further classified to pre-obese, obese class 1, class 2, and class 3. So the reason why it was done like this, because, at the higher BMI of at 23 kilogram per meter squared and above, it is associated with increase in mortality. Lots of studies around the Asian region have shown that, associated with increased incidence of diabetes, pre-diabetes, and also cardiovascular disease. So the burden of obesity in Malaysia has increased, and there has been a clearly documented dramatic increase in the prevalence of obesity in Malaysia over the last 3 decades. And it is termed nutrition transition because of the availability of calories, mainly in the form of plant products, to diet that high in fats, sugars, and energy-dense processed food. So in our population now, there's a lot of processed foods is easily available in the supermarkets, in the shops, and even at school. And with this movement control order, because of COVID pandemic, this food availability of food, processed food is even a lot more, because people bought or buy the foods and keep at home, worried that there'll be problems getting foods when during the MCO. And this change in available calories, fat, and protein per capita in Malaysia has started many years ago, as long as 40 years ago. And if you can appreciate here that in the last 45 years, the fat and sugar intake has increased by about 80% and 33% respectively. And the change in overweight and obesity prevalence in some Asian countries is actually comparison to the United States back then. So you can see here in looking at this graph, for Malaysia, it has increased slowly, this is until 2010, but I'll show you the latest one. And if you look at where is Malaysia in the region of Asia Pacific compared to Australia, UK, New Zealand, and even US, there, this is where we are, okay? Being Asian with a small body frame, the population or overweight population is increasing dramatically, and you can see is keep on increasing and almost reaching to the Western or Caucasian population with a bigger body frame. So in Malaysia, we've done a lot of obesity studies. There's a large-scale studies dated since 1996. In 1996, we have NHMS National Health Morbidity Survey, the second, whereby at that time, we still use a WHO cutoff point, whereby overweight is classified as 25 kilogram per meter squared and beyond. So overweight is 16.6. Imagine if I bring it, if I use the Asian cutoff point that's been introduced a couple of years ago, then the prevalence of overweight will be even higher. So, but then it was 16.6, obesity is only 4.4. And in another study involving a 10,000 population, the prevalence of overweight has increased within short period of time, within about 7 years to 26.7%, and obesity 12.2%. And in 1996, if you look at prevalence above 20 years, prevalence among those aged 20 years and above is 20.7, and 16.6% is actually 30 years and above. So this is 30 years old and above. This is 20.7% is for those beyond 20 years old. And another study after that, again, another big population looking at obesity has increased further to 11.7%. And again, even this is above 30 years old. If you look at above 30 years old, it's 12.6% prevalence of obesity. And again, all these prevalence based on WHO criteria. NHMS, the third in 2006, even bigger number of participants, more than 33,000. The prevalence of overweight has increased further, and similarly obesity to 14%. We did our MSSM study, metabolic syndrome study, 4,000 population, whereby we also perform OGTT in these 4,000 subjects. The prevalence of overweight and obesity has increased again, 33.6% and 19.5% respectively. And in 2011, another National Health Morbidity Survey showed 29% of overweight, 29.4% of overweight, and 15.1% prevalence of obesity. And if you look at the prevalence of overweight and obesity according to gender, you can see that women basically had a higher prevalence of obesity as compared to men, okay, in general. But with regards to overweight, there's not much difference between men and women. So I stopped short at 2011 National Health and Morbidity Survey. This is another another graph or another data showing more information about that further on or later on when National Health and Morbidity Survey was done 4 years later in 2015. So the red bar represents 2015. You can see that prevalence of overweight are not much change compared to 2011, but prevalence of obesity has increased further to 30.6%. And what about obesity among school children? So in 2005, 3,000 school children aged between 13 to 17 years old in urban area, overweight and obesity 11.4% and 8.2% respectively. 2008, 3 years later, about the same age group, smaller scale study, semi-urban, you can see the prevalence of overweight has increased. Similarly, prevalence of obesity increased further. So with modernization and development, the weight of our population increased more and more. 2010, similarly, in urban area, more than 1,000 subjects, school children were looked into, or adolescent, 13 to 17 years old, overweight 12.5% and obesity about 11.7%. And about 4 years ago, adolescent, again 6,000 school children, semi-urban, 16% overweight and obesity 11.5%. And back to MSSM study where we did OGTT in this population. Like I mentioned, overweight 33.6%, obesity 19.5%, more in females, okay, 22.5% as compared to males. And the incidence of newly diagnosed diabetes is found to be increased by threefold in those who had BMI more than 30 kilogram per meter squared. So it's not just about BMI and weight alone. It also associated with increase in comorbidities or increased prevalence of diabetes. So if we look further in the same study, BMI less than 18.5, higher percentage of non-diabetic. But of course, as the BMI increase, you can see more and more. Okay, you can see even beyond 25, a lot more, and this is between 18.5 to 24.9, still according to WHO criteria, not the Asian criteria. You can see 68.9% non-diabetic, diabetic 8.4. But as the BMI increase further, you can see the prevalence of IGT increase from 11.9% to 25.4%. And these are the people or participants or subjects who will then develop diabetes if we don't do anything to the BMI here. Okay, and similarly, IFG is increased as the BMI increases. So what actually contributed to the increase in BMI or increase in obesity among Malaysians? So, people talk about sugar tax, reduce the prevalence of diabetes, obesity, because then people will, population will take less sugar and so on. Basically in Malaysia, the food culture is the one that bond people together over a variety of meals. People like to meet up, drinks, and eat together. And 36% of Malaysian students have carbonated soft drinks at least once a day. And they also take heavy meals after supper. There's a 24-hour restaurant being opened before the MCO or during before the COVID pandemic, COVID-19. And 70% of Malaysian adolescents consume heavy meals after supper, like I mentioned. And this is not just once a week, it's between 1 to 6 days in a week. So in many occasions, they will take heavy meals after supper. And 7 in 10 adolescents skip breakfast regularly, and therefore that leads to heavy eating whenever they feel hungry at lunch time. And this leads to about 74% of obese adolescents actually skip breakfast. And the same food culture, like I said, eating together and so on, has led to 13.3% of Malaysians become obese. And we become the highest among in regards to obesity among Southeast Asian countries. So in recent 2019 NHMS have showed that the rising trend in weight gain and about 50.1% of adults were actually overweight or obese. So 30.4% were overweight and 19.7% were actually obese. And again, 54%, 54.7% were women, and especially those who aged between 55 to 59. And during this time, they start to have also arthritis, back pain associated with obesity. Therefore, with regards to exercise, healthy lifestyle, there's a lot of limitation for them. And 2019 NHMS finding also showed that 29.8% of children between 5 to 17 years old were overweight or obese. And of course, again, with the recent finding, Malaysia became the highest country in the Southeast Asia with regards to adult population who were obese, with prevalence of 15.6% comparing to other neighboring country. And recently in February, not very recent, 2020 February, WHO has has stated that obesity has reached epidemic proportions globally, and a lot of people obese or overweight. And in Malaysia, because I run the obesity clinic, the condition worsens with MCO or movement control order because of COVID-19 pandemic. And this is from the economics intelligence unit about 4 years ago. The percentage of overweight population with a BMI more than 25 standardized age standardized to adjusted estimate that adults in Malaysia are obese. You can see overall about 37%. Of course, female again is the champion. We are again leading leading population with obesity and overweight as compared to other neighboring country. And if you look at the total cost of obesity as a percentage of healthcare spending, you can see that 19.36% of is actually need to be spent to treat obesity and associated diseases associated with obesity, much higher as compared to other countries around the region. And if you look at the total cost for obesity as percentage of nominal gross domestic product, obesity also has a direct impact on the country's GDP performance as shown in this figure. So Malaysia's total cost for obesity as percentage of nominal GDP ranks top at a range between 0.4 to 0.8%, far ahead as compared to other Asian countries. So really there's a lot need to be done with regards to obesity in Malaysia. Of course, studies and a lot of prevention still need to be done, even at earlier stage at the when they are much younger, children, adolescents, because they go on and have a problem with obesity as they go to their adult life. Thank you. I welcome any question.
Thank you, Prof. Laila. Very alarming data from Malaysia. And my apologies for the misinformation earlier. Prof. Nolaila is the current Deputy Dean for Postgraduate Studies in the Faculty of Medicine. So, right, here are two interrelated questions for Prof. Laila from the audience. Can we reverse the diabetes status by controlling our weight? Can the diabetic drug helps for this purpose?
Right. Good question. Thank you, Shopang. Well, if let's say the reason why somebody developed diabetes is due to obesity, yes, by losing weight, that person should be able to reduce blood sugar. And if it's very early stage whereby the beta cell is still functioning very well, there's no beta cell apoptosis, I would say yes, would be able to, but still we would continue following up the patient yearly, because if let's say the patient has a risk with regards to gene in developing diabetes and so on, we still have to monitor and look at the blood sugar somehow or other. With aging also, our blood sugar tends to increase slowly, about 0.1 millimole every year after the age of 40. That's what I was told. So despite normalizing blood sugar with weight loss, we should continue monitoring our patient. And whether anti-diabetic agent would be able to reverse this, yes, nowadays people talk about GLP-1 analog. With GLP-1 analog, subject or patient should be able to lose weight, and with that also control the blood sugar. But again, you know that pathophysiology, that's multifactorial. Okay, I would not be able to say that yes, it sort of cure diabetes totally. There's also other contribution or other factors contribute to hyperglycemia, insulin resistance, beta cell dysfunction. We know very well that hyperglycemia because of the SGLT2 receptor in the kidneys as well. So the more we know, the more careful we will be with regards to saying that, yeah, your diabetes is cured. What important is to be follow up or monitor this patient closely. Because again, obesity is a disease. So once it's a disease, you have to monitor the patient forever. Similarly with diabetes. I do hope that answered the question.
Thank you, Prof. Laila. We will allow more questions at the final session, yeah? So, right, thank you, Prof. Laila. We will move to
Thank you.
the next speaker. So we would like to welcome Dr. Jennifer Carter from the University of Oxford. So Dr. Jennifer is a Senior Research Fellow and Associate Course Director for the Master of Science in Global Health Science and Epidemiology at the University of Oxford. She will talk on obesity and cardiovascular risk, a comparison of the UK and Malaysia. Please welcome Dr. Jennifer.
Hi, great. Thank you very much. Can you see my slides okay?
Yes.
Okay, great. Great. Well, today I'm going to present some initial results that we've started getting out from this collaboration between the UK and Malaysia, and looking at the relationship between obesity and cardiovascular risk. Now, one of the questions that we were interested in investigating with this collaboration was to look at the relationship between body composition and heart disease across different ethnicities. And as Sarah presented earlier, some of the inspiration for this came from these very striking results that we were getting out of Chennai in India, where when we were looking at the relationship between BMI and cardiac mortality, as you can see, the relationship was very flat. So as participants became overweight and obese, we didn't see any increase in the risk of heart disease. And this was very much in contrast to the J-shaped relationship that we see consistently in Europe and North America and other places around the globe, where you get this increased risk at lower BMIs, and then you have this strong positive association as people gain more and more weight and become overweight and obese. So we really didn't understand what was going on with what we were seeing in India. As well, some initial results out of the Malaysia cohort was looking at the relationship between obesity and diabetes. And again, we're seeing some interesting findings. As we can see on the left panel, this is the percentage of participants that are obese as measured by BMI. And we could see that the Indian and the Malaysian participants had equivalent levels of obesity. But when you look at the right-hand panel, which is the percentage of participants that were diabetic, the prevalence in the Indian subpopulation was nearly double that of the Malays at every age. So by the time we got to age 80, about 20% of the Malays had been diagnosed with diabetes, and this was nearly 40% in the Indian subpopulation when they had similar levels of obesity. So this really made us want to understand in more detail what's the potential relationship between obesity and disease across these different ethnic groups. So this collaboration between UK Biobank and the Malaysian cohort was begun. And for the UK Biobank, if you're unfamiliar with it, it's a very large cohort of about 500,000 adults around in their late 50s. And recruitment was begun back in 2006. And they have a huge breadth of measurements on lifestyle, physical measurements, health, and blood samples. So it's a fantastic resource. If there's one limitation of UK Biobank, as you can see, is that it's about 95% white, which makes looking into relationships between ethnicity and disease very difficult. Now, the Malaysian cohort, which we'll be hearing about more later today, is a very large cohort of about 100,000 individuals, a little bit younger than the UK Biobank participants, but recruitment was begun around exactly the same time. And similar to UK Biobank, they have a large range of measurements on lifestyle, health, and blood samples as well. And one of the beauties of the Malaysian cohort, as you can see here, is they have large numbers of Indian, Chinese, and Malay participants. So it's really this perfect complement to UK Biobank, where we can start to look at questions about the mechanisms underlying disease very reliably in large numbers of participants across different ethnic subpopulations. Right, so the first analysis that we've been working on is investigating the relationship between body composition and risk factors for heart disease and diabetes in the white Europeans in the UK versus the Malay, Indian, and Chinese populations in Malaysia. Now, body composition is thinking about the different components of the body in terms of fat mass and lean mass and where that might be distributed. And we had several measures that we were looking at in terms of body mass index or waist circumference. And we had several risk factors for disease that we've been investigating. Just for today, I'm just going to be presenting information looking at body mass index, fat mass, and lean mass with systolic blood pressure and LDL cholesterol and triglycerides. And as well, I'm just going to be showing the results that we've seen in men. One thing to note is that our measures of body composition and our risk factors for disease, these were all measured cross-sectionally at the same time at baseline in these two studies. So a few characteristics of our participants. If you look at the first column with UK Biobank, you can see that we're just larger in the UK. So we have more lean mass. Our fat mass is actually pretty similar, but we're about 10 centimeters taller. We're about 13 to 16 kilograms heavier. Our BMI is a little bit bigger, but our waist circumference is much wider, as are our hips. So again, really interested in starting to look at what these differences in body size, how it might relate to risk factors for disease. Okay, so first let's go ahead and look at the relationship between body composition and systolic blood pressure. What we have here on the left, we have the four ethnicities. And I've presented two linear regression models. So the first model is my basic model. So this is adjusted for age and height, education, and a variety of lifestyle confounders. And my second model is mutual adjustment. That means that I've added in an adjustment for other measures of body composition, because measures of body composition are highly correlated. And what we wanted to see is what's the relationship of BMI, say an overall measure of body size, independent of, say, your waist circumference. So irrespective of the fact you might have more adiposity around your waist, what's the relationship of BMI to your systolic blood pressure? And what we can see here is that in the UK, about 4 kilograms per meter squared higher BMI was associated with about 3 millimeters of mercury higher systolic blood pressure. And this was generally equivalent across the four ethnicities in the study. When we move on to fat mass, what we saw in UKB or UK Biobank, we had a slightly weaker association with fat mass than we did with the 3 ethnic groups in the Malaysia cohort. And when we looked at lean mass, and this is similar to muscle mass, we could see this very slight positive association where you had a slightly higher lean mass was associated with slightly higher systolic blood pressure. And it might have been a bit weaker in the Indian subpopulation, but generally equivalent within the other ethnicities. Okay, let's go ahead and look at LDL cholesterol. And with LDL cholesterol, what we saw with BMI was that we had weak positive associations, and they were generally equivalent across the different ethnicities in the study. When we looked at fat mass might have been, looked like it was a bit stronger, had a bit of a stronger association with LDL cholesterol than BMI did, particularly in the UK and the Chinese populations. And when we looked at lean mass, what was interesting is that the association flipped from null to inverse. So when we adjusted lean mass for fat mass, we saw that lean mass appeared to be protective. So higher lean mass was associated with a lower LDL cholesterol. And when we look at these relationships with fat mass and lean mass, whilst they were somewhat equivalent, pretty similar between the ethnicities, you can see that BMI is really not telling the whole story of what we're looking at here between body size and risk factors for disease. And lastly, I'm going to look at triglycerides. And when we looked at triglycerides, what we noticed was in the Indian subpopulation, the results appeared to be a bit weaker than they were for the other ethnicities. And when we looked across all the results as well, we could see that fat mass appeared to have a stronger association with triglycerides than BMI. And this was really apparent when we looked at the Malaysian subgroup. So in the Malay participants, there was no relationship between BMI and triglycerides, but we had this positive relationship with fat mass in the Malay participants. And lean mass had a very weak positive association with triglycerides, but again, generally equivalent across the different ethnic groups. So in summary, we had a lot of similarities between the ethnic groups. For systolic blood pressure, fat mass might have been a bit weaker in the UK, and lean mass might have been a bit weaker in the Indian subgroup. For LDL cholesterol, again, generally similar associations. Fat mass appeared more important than BMI, and lean mass had a protective inverse association. And triglycerides, we saw that fat mass again was more important than BMI for all of the ethnicities, especially in the Malay participants. And as well, we saw that BMI and fat mass were weaker in the Indian participants. So strengths and limitations. So I think overall, hopefully you can all appreciate this has to be one of the largest comparisons across ethnicities of adiposity and risk, probably around the globe. Very, very few people have these measurements on such a large groups of participants. So it's really exciting to start looking at these associations more reliably with so much data. Another strength that's fantastic between the UK Biobank and the Malaysian cohort is they have very similar measurements, including the time period when they were taken. It should be noted though that fat mass and lean mass were measured with bioelectrical impedance analysis, and the machines that measured these differed across the two cohorts. So we are going to be looking into more depth about how that might be impacting what we're seeing. As mentioned, these are cross-sectional analyses. All the measurements were taken at the same time, so we can't rule out reverse causality. So it might be that people who had subclinical disease, which would be related to their risk factors like blood pressure and lipids, could have also been impacting their body composition. Okay, and in conclusion, generally we saw a lot of similarities across the ethnicities in what we've looked at so far. And I think what this is really leading us to is that what we've seen here doesn't seem like it's going to fully explain the differences that we see between BMI and disease that I've presented earlier in the slides of my presentation, and which you saw in Sarah's presentation as well. And it really suggests that we need to have better, more in-depth measurements of body composition. And so this is leading on to what we're doing next, which is ASEAN's going to be presenting later on in the webinar today, is that we're now doing DEXA imaging scans to really get better measurements of where the body fat's distributed across the body, and then looking at this relationship to disease. Hopefully trying to get an idea of what's a healthy range for body composition in Malaysia, and should public health guidance on obesity, should this potentially differ even more by ethnicity. Okay, and I say thank you to the Oxford team, particularly Hannah and Paul, who helped a lot with the analysis and the slides. And then a big thank you to the Malaysian team, because every time we changed the analysis, we only had to do it once for the UK, and the Malaysian team had to do it 3 times for all the different participants in their study. And they've been really helpful and really lovely to work with on this collaboration so far. So thank you very much.
Thank you, Dr. Jennifer, for the interesting data. So here is one fresh question for you from the audience. Although DEXA may be a better measurement for the body composition in assessing the risk of non-communicable diseases, how are we going to apply this into clinical practice, since DEXA is not that cheap and requires special facility compared to the standard anthropometric assessments such as BMI and waist-hip ratio? Any comment from you?
Yeah, I mean, it's a really good question. And I think the first step is we need to understand the mechanisms of disease a bit better. So, we need better measurements to understand what's potentially going on with body composition and disease, particularly in different subgroups of the population. And once we understand that better, then we can start to look between, we can look between this relationship between, let's say, gold standard measurements and more feasible, cheaper, practical measurements that you can do in the clinic. So what's the relationship between what we see on DEXA and say what you would get from measurements of waist circumference or measurements of waist to hip ratio, hip circumference. So I think first you understand the mechanisms well, and then you can see how you can scale up the implications from that into something that we could feasibly measure and implement in practice.
Right. So thank you, Dr. Jennifer. We will allow more questions for Dr. Jennifer at the final session. To the audience, keep posting your questions at the Q&A box. Some questions already answered by our panelists. All right, so next we would like to welcome Dr. Carmen Piernas, also from the University of Oxford. Dr. Carmen is a university research lecturer in the Nuffield Department of Primary Healthcare Sciences at Oxford. She will speak on dietary approaches to obesity. What works? Please welcome Dr. Carmen.
Thank you so much. So the focus of my presentation today is completely different. It's on diet. So I'm going to be discussing some evidence about diets and nutrients on obesity. In the interest of time, I'm only going to present obesity as an outcome. I'm not going to be talking about cardiovascular disease or diabetes, but we can talk about it later during the panel discussion. So I'd like to start with this picture, which is the obesity system influence diagram. This is a project conducted a few years ago now, where you can see all possible connections and interactions and pathways which relate to obesity. And this is just to show you how complex obesity is. And I would like you to keep that in mind when we talk about diet and how difficult for people is to reach and maintain a healthy weight, because it's a very complex system, and we can't just tweak a few things and pretend people can lose weight that easily. But without trying to oversimplify, I would like to point out a few important things that we know for sure. And that is that weight gain arises from chronic positive energy imbalance. Most people are less active than what they should be for good health, and most people eat more than they need. So energy intake is truly a fundamental determinant of obesity. but it's not the only thing. And there are many things that determine energy intake. Therefore, solutions need to account for the complexity of this system. So with this picture in mind, I'm going to be discussing evidence around nutrients. For example, is dietary fat, is the fat that we eat in our diet extra fattening? Well, there are reasons to think so, because dietary fat contributes a lot more energy. So there is 9 calories per gram of fat when we eat dietary fat. So it's more energy dense, it's also digested easily, more easily absorbed, more easily. The diet-induced thermogenesis of fat is also lower. It's also oxidized after other nutrients like carbohydrates. And it's also like adding extra palatability to the food. So foods that are high in fat are also highly palatable. So are more likely to be eaten in bigger amounts. So there are reasons to think that fat is extra fattening. And this is a meta-analysis conducted a few years ago now of lower fat diets compared to usual fat diets on body weight. And there is a lot of studies with a lot of heterogeneity. The overall point estimate shows that a lower fat diet overall results in about 1.5 kilograms less body weight compared to the usual fat diet. And these differences in mean weight loss is bigger when the difference in percentage of body fat is also larger. But how about carbohydrates? And there's a lot of debate more recently about carbohydrates, especially about the quality of the carbohydrates. The Scientific Advisory Committee for Nutrition in the UK has reviewed all of this evidence. And in general, when it comes to body weight, the evidence is actually very limited and very inconclusive for most of the whole grains, fiber, and glycemic index. Except for some limited evidence on energy intake. But overall, the evidence on body weight is actually rather limited. However, there is good evidence on free sugars. So this is a meta-analysis by Temorenga in the BMJ of the effect of adding sugars in diet. So diets that increase sugars among adults have found in short-term studies about 0.5 kilograms more. For longer terms have found 2.73 kilograms more weight, and the overall effect's about 0.75. So diets that have higher sugars in ad libitum diets in adults tend to increase body weight. However, when we see the isoenergetic replacement of sugars with other carbohydrates, we have no effect in short or term or the overall effect. So the effect of sugars, again, like it happens with dietary fat, it happens through the effects on energy intakes. Finally, the effects of protein. Protein tends to be more satiating than other nutrients. It's got higher diet-induced thermogenesis as well. And it's been associated with spontaneous undereating. This is a trial conducted quite a few years ago now, and we can see the high protein, low GI diet is the one associated with weight change, like decreased body weight compared to all these other diets. So in general, can we say that diets with different macronutrient contents can achieve different changes in body weight? Well, this is again a trial in 2009 in the New England Journal of Medicine, testing different diet compositions in terms of carbohydrate, protein, and fats. And as you can see, at every time point, the differences in weight, in changes in weight from baseline are very minimal. So all of these different diets with different macronutrient compositions are achieving very similar changes in body weight at every time point. So what matters really is dietary adherence, is more important than the macronutrient prescription. As we can see here, this is another trial, quite old now, where we can see different types of diets like Atkins, Zone, Weight Watchers, or Ornish. They all achieve significant weight losses at 12 months. And what we can see here is a very strong association of dietary adherence with any type of diet achieving a larger weight loss at 1 year. So in this context, we, as a recap, we can say that macronutrients are important. They've got some effects, but what matters really is adherence to any of the diets. What I've shown so far has all been dietary intervention studies. I'm going to present now some of the observational evidence that we have been doing in the UK Biobank study with diet. For example, we looked at the adherence to WHO dietary recommendations. For example, less than 10% of saturated fats, 10% of free sugars, more than 25 grams of fiber a day, and more than 5 servings of fruits and vegetables a day. And in terms of weight, we found strong trends for lower BMI, total body fat in percentage, and waist circumference among people who are meeting more of these dietary recommendations. As I said, I'm going to discuss only body weight. We can discuss later about cardiovascular disease, but we found, for example, in the same study that we're preparing now, we found increased risk of mortality and CVD death among people who are meeting less of these recommendations. And then secondly, this is a study we have conducted on dietary patterns in the UK Biobank, where we used reduced rank regression, looking at patterns that explain energy density, free sugar, saturated fat, and low fiber. And one dietary pattern that emerged very clearly, high in chocolate confectionery, butter, low fiber bread, and low intakes in fruit and vegetables, again found a clear linear trend with BMI. So people scoring higher with this dietary pattern had a higher BMI. Again, this study found increased risk, very clear linear trend in risk in cardiovascular disease and diabetes, which is mediated by BMI. So in terms of what works for obesity, I'm going to be showing some evidence of meta-analysis that we have conducted in our department. So this is the effect of behavioral weight management interventions. So Weight Watchers or similar weight programs where people receive, for example, education and different behavioral techniques to change their diet and lose weight. And overall, we find that behavioral weight management interventions versus minimal controls at 12 months achieve a small but significant amount of weight loss, about 2.84 kilograms. There's a lot of heterogeneity in these studies, as you can see, because they achieve weight loss through many different ways. But we also conducted a meta-analysis of meal replacement diets. So this is when people diet using shakes and bars and soups, for example, and they replace a whole meal with these kinds of things. And what we showed, for example, at 12 months, a meal replacement diet compared with to the standard diet only can achieve an extra 1.44 kilograms of weight difference. But if we compare a meal replacement diet with extra support versus a conventional diet with extra support, the meal replacement diet can also achieve an extra 2.2 kilograms on top of the weight loss that you would achieve normally. So meal replacement diets seem to increase a tiny bit more the amount of weight loss that you can achieve. And finally, this is a meta-analysis of very low energy diets. So these are typically diets that require people eating less than 800 calories per day, usually in liquid form. And these diets have found at 12 months compared to, so a very low energy diet plus a behavioral program compared to just behavioral program has been able to achieve an average of 4 kilograms extra weight difference compared to just the behavioral program. So as you can see, a commercial weight loss program with, low, let's say like minimal, or what we say, oh gosh, I forgot the name now. Low intensity programs can achieve about 2 to 3 kilograms. And these meal replacement diets or very low energy diets can enhance that amount of weight loss. So this is just finally to remind you of the diabetes prevention program in the US. This study, as you can see, at 4 years, people regained their weight. But what matters is that even a small amount of weight loss brings surprisingly large health benefits, as you can see in the bottom graph. So even when there was weight regain in the long term. So in summary, macronutrients are important and they have small but measurable effects on body weight. However, what we know, and we've got good evidence, is that an energy-dense, high-fat, high-sugar, low-fiber diet increases the risk of weight gain. Weight loss requires a calorie deficit, and long-term adherence to a hypoenergetic diet is a key determinant of success. And long-term weight control requires permanent changes in eating habits to prevent regain. However, even with weight regain, small weight loss leads to significant long-term health benefits. So it's worth trying. And that's everything from me. Thank you so much.
Thank you, Dr. Carmen. Very interesting findings. I'll read out one questions from the audience for you. With regards to the high percentage of overweight people skipping breakfast, would you suggest intermittent fasting as a strategy? What is your take on that?
Well, intermittent fasting is now becoming quite trendy. So I don't think there is enough research to support one strategy over the other. So I suppose intermittent fasting, if you can adhere to intermittent fasting for a long enough period of time that allows you to keep doing it for a long time, it's going to be effective for sure. So anything that you can stick to would be effective as long as you do it. People is also concerned about breakfast. Is breakfast good? Is breakfast bad? Well, people who have really bad breakfast, well, they should probably skip it if they are having high sugar, high fat things. So having a high sugar, high fat breakfast is a bad idea. So it's better to skip that breakfast. But if you're gonna have fruit and whole grains, it's probably a good idea to have breakfast.
Mm-hmm. So the key is always to be consistent.
Exactly. I think the evidence suggests that any diet works if you can stick to it in the long term.
Okay. All right. Thank you, Dr. Carmen. So as usual, we will allow more questions at the final sessions. Right. I guess we will take a 10-minute break, and according to the schedule, we will take a 10-minute break and we will be back at 5:25 p.m. Malaysia time or 9:25 a.m. UK time for another interesting talk by Professor Frederick. So please stay tuned. Hello, welcome back. As a friendly reminder to the audience, you may post your questions online at the Q&A box, and we will highlight it to the respective speaker to be answered online. We will try our best to allow 1 or 2 short questions immediately after the talk by each speaker. But for those that your questions were not read out during that session, don't worry, we will also have a final Q&A session after the last speaker. So we will try our best to highlight your questions either in the first or the final session. Okay, so our next speaker is Professor Frederick Carpe. Professor Frederick is a professor of metabolic medicine and consultant physician at the Oxford Center for Diabetes, Endocrinology and Metabolism. He will speak on the use of genomics to gain insight into functional properties of upper and lower body fat depots in humans. So let's welcome Professor Frederick.
Thank you so much. It's a pleasure to take part in this symposium. So fat distribution has been alluded to by several speakers so far, and I will give you just some little insight of how I can go about finding out function. That is also something that has been mentioned several times, which we can't really get from the epidemiological associations. So I'll start off by the simplest of all examples, where there's a genetic defect leading to a substantial effect on fat distribution. And this is the example that has been known for decades, consisting of familial partial lipodystrophy. These are rare cases, and this particular case has Dunnigan-Coberling syndrome with a mutation in the LMNA gene. And as you see from this woman's legs, they are muscular, but they are completely fatless. There is no subcutaneous fat whatsoever in the lower body as part of this syndrome. And this has enormous consequences, enormous metabolic consequences. This particular patient is only 29 years old. She has had a mini stroke, TIAs, triglycerides of 7.2 millimoles per liter, a fatty liver. So there are substantial metabolic problems that you have here. And this gives inspiration to understand what functions the different fat depots have. Could it be that it's actually a major determinant of the whole body metabolic milieu of how the fat tissues work in different ways across the human body? So we have spent a lot of time over the past 2 decades to actually understand these issues. We have conducted a range of physiological studies. We are focusing nowadays more on cell biology and genetics. And I'm just showing you a cartoon here to illustrate some of the key findings we have had so far, where we know nowadays that the progenitor cells in abdominal and gluteofemoral fat tissue are very different. They respond differently to the requirements for fat storage. The upper body cells respond with rapid fat storage and also rapid fat release triggered by stress hormones, whereas the lower body fat storage is really there for a very long term, very intrinsically related to evolutionary processes and a safe storage for fertility, not least. And these signatures in the lower and upper body fat stores are very different. So these cells are share the same genome, but they express different proteins and different genes, and that gives the different characteristics. And I'll give you one physiological example that is particularly striking, really demonstrating how important it is, how clear it is that these fat depots are so different. So this is a study we conducted some years ago where we looked at the principal function of adipose tissue, that is to release non-esterified fatty acids. And in black dots here, we have the output of non-esterified fatty acids when we give systemic adrenaline into human beings. And you see as a dose response, there's a very robust lipolytic response with massive release of non-esterified fatty acids from the abdominal tissue in the individuals. Whereas in the same individuals, if we look at the outflow of non-esterified fatty acids from the gluteofemoral region, it doesn't respond at all. So these tissues are engineered to do very different things to different responses. So how can we then understand better how the tissues actually work? So the inroad I'm going to describe and give you just a couple of examples of is either how we use genomics to understand cell and tissue phenotype, or what I will spend more time actually on is to talk about phenomics, how we use phenotypes to distill out which genes to look for to understand function. As I started off with my slide on familial partial lipodystrophy, that is of course the crudest example you can think of. A monogenic disorder gives a very strong phenotype. And ironically, we still don't understand and have no good tools to understand how the coding changes in the LMNA gene actually give rise to a specific fat loss in the lower body. So that is still an outstanding question. So one inroad, if we start with genetic variants and try to understand gene function in adipose tissues, is to use genome-wide, the hits from genome-wide association studies that were conducted looking at fat distribution. So this is the large studies that has used waist and hip measurements adjusted for BMI and come up with a number of loci that associate with fat distribution. And I'm not going to elaborate much on this at all, but we have taken a number of the candidates that come out from these very robust and large studies and used cellular models to understand function. And that has indeed delivered very nicely. And we do understand now how certain genetic variants actually are relevant to certain tissues for fat storage or just to produce progenitor cells for expanding a tissue. What I'm going to turn to now instead is the other approach, and that is what we call phenomics. And I'm going to show you first a paper that I think is really worth reading, a very intelligent study by Luca Lotta from Cambridge, where they used an integrative genomic analysis and came up with adipose tissue fat storage based on human insulin resistance and related phenotypes. So they used a large number of different cohorts, but fasting insulin or insulin resistance was core in these phenotypes. They also included raised triglycerides, HDL, and insulin sensitivity. They used more detailed effects on fat distribution like DEXA to understand fat distribution. So I won't go into details, but what they ended up with was a large number, actually 53 gene variants that showed a very good association with this combined metabolic syndrome or similar type of phenotype constellation. And they are in this heat map graded according to the actual association with insulin sensitivity. So since insulin sensitivity was what the investigators actually looked for, I'm sure they were quite intrigued to see that 5 out of these 53 genes actually mapped directly into the insulin signaling cascade at different steps of the signaling pathway. So that was a good find. I suspect that the investigators didn't anticipate what they found, as I think was the even more intriguing and interesting observation. And that was if they took all these 53 gene variants associated with insulin sensitivity, raised triglycerides, low HDL, etc., and looked at the association using a gene risk score of fat distribution. It turned out that if you have a high gene risk score, many of these gene variants, you have a much lower mass of your leg fat. So it was a very, very clear dose-response relationship between the gene score for the metabolic syndrome and having a lower fat mass in the legs. So it gives a really interesting association and confirmation at the population scale to the partial lipodystrophy concept that I started with. So one of the genes that came up in this long list was a gene that had very little functional annotation called KLF14. And it actually turned out that we were working on this gene already when this paper was published. So this really it started to give a lot of insight to what was going on here. And the inroad for us here was actually that KLF14 is also a diabetes risk gene, but it is not amongst diabetes risk genes as we think of them in common. It is actually the rare cluster of genes that are associated with insulin resistance rather than poor beta cell function. So how could it be that a gene expressed in adipose tissue is causing a diabetes risk and it's mediated through insulin resistance? So there are quite a few steps there to understand, but since it's nearly exclusively expressed in adipose tissue, that's the place where we have to start to look for it. So Mariana Todorovic, one of my postdocs, has devoted a lot of time to studying this particular gene and its function in human adipocytes and human adipose tissue. And I'm just showing here one of the tools that she has used, and that is to use subcutaneous abdominal biopsies where we can prepare progenitor cells and use knockdown models of KLF14 to study its function in vitro. I'll come back to this, but first I will show you some cross-sectional data that were very striking we got early on. So these were from the same biopsies that we took. We also looked at the cell size, adipocyte cell size, according to genotype. So in the Oxford Biobank, in the top of the slides, we have the capability of recalling people according to genotype. So we recalled people according to who are homozygous carriers for the non-risk allele, the T variant of the KLF14 that otherwise give diabetes risk. And you see here that in women, you have this very, very substantial effect compared with the CC variant, that is the diabetes risk variant, on average adipocyte size. Normally, adipocyte size is something that tracks extremely well with overall adiposity, but here we had matched people for BMI, so that was no longer an issue. This is a genuine effect directly by the KLF14 gene. And the effect was so striking, but in the Oxford Biobank only illustrated in reasonably small numbers. So we were a little bit cautious about the interpretation and therefore asked the collaborator in Sweden, Peter Arner, who had a larger collection of adipose tissue biopsies that had been cell-sized. So we went back to his cohort and genotyped it for KLF14, and sure enough, could reproduce the same finding, again, using completely different techniques in a different cohort, but asking the same question. So how can we square this, that we have a phenotype that is structurally very different according to genotype in adipose tissue? How does the function in adipose tissue, how is that affected by KLF14? So again, we were helped by Peter Arner's old data, where he had been looking at lipogenesis using radioactively labeled glucose and looking at the incorporation of glucose into triglycerides in isolated adipocytes. So taking his genotyped cohort for the KLF14 risk and the non-risk variants, the TT and CC, we could see that the incorporation of glucose for the risk variant was much lowered in women. Nearly half of the glucose was taken up if you had the diabetes risk variant. So it was clearly an aberration in lipogenesis in these participants with the diabetes risk variant of KLF14 in adipose tissue. We then turned into the model I initially described that Mariana Todorovic set up, where we had an in vitro system simulating what the genotype is doing in real life. So the risk variant actually leads to lower expression of KLF14. So if we have an in vitro system and knock down KLF14, we simulate the effect of the diabetes risk variant. And you will see here, this is a successful experiment where we had about 50% knockdown during an adipocyte differentiation curve of KLF14. And this has consequences, actually not for the typical markers of adipocyte differentiation, but there is clearly less of several very important functional partners in adipose tissue, such as expression of leptin, and perhaps most notably expression of GLUT4. And that is beginning then to explain possibly why there was such a low glucose uptake and low lipogenesis in the carriers of the diabetes risk variant. So moving on to see what happened with the adipocytes, not just the expression, we looked at three dimensions of function. One was proliferation of pre-adipocytes. So we take the progenitor cells from biopsies, we look at the effect of knockdown, and in the knockdown cells, it was a very paradoxical result. So here's the doubling time. So if the doubling time decreases, that means that the proliferation actually increases. So that is slightly paradoxical, that you have a tissue with larger cells, but you have increased proliferation. I'll come back to a possible interpretation of that. What was very reassuring was to look at lipogenesis, and this is lipogenesis conducted in a very different way compared with what I described before. Before the cells, mature cells, were exposed to radioactively labeled glucose to look at the incorporation into triglycerides fat. Here, we look at the total accumulation of lipids in differentiating adipocytes in vitro. And you see, again, about 50% less triglycerides were accumulating if the KLF14 was reduced, expression was reduced in the knockdown model. And finally, glucose uptake was much reduced as well. So in this in vitro system, we actually confirm exactly what happened in the ex vivo system using mature adipocytes. So let me summarize. Human fat depots, they have different fat storing capabilities, and this contributes to the whole body metabolic profile. And studying using a candidate gene approach from GWAS studies, we think we see that the regulation of discrete fat depots, expansion of function, is very complex, and it's definitely driven by polygenic traits. And using these follow-up of GWAS studies, it's quite clear that fat distribution seems to reveal specific processes. It's not only lipogenesis, It's sometimes proliferation, and sometimes it's a combination of the two. And it looks as if each distinct fat depot has its own regulators. And that's why we have this enormous range of small effects that gives this nearly gene score effects over a large continuum of fat distribution that we see in populations. I'm very happy to take any questions at this point in time.
Thank you, Prof. Frederick. Thank you for sharing such an interesting story of KLF14. I do have a question from the audience for you. What would be the rationale to investigate at the genomic level since they are unmodifiable?
I think it depends on what you mean by unmodifiable. Was the question modifiable or unmodifiable?
Unmodifiable.
Unmodifiable. Well, I think it's a very interesting question. I think I would probably have an issue with unmodifiable because they may be unmodifiable in terms of when they have been established. Assume that they were established very early on in growth and development. Perhaps then it's a trait that we live with for life. However, there is probably a time window when they are modifiable at earliest growth and development. So I'm intrigued by the possibility of looking at maternal nutrition, maternal health, to look at the springs, body composition, and even fat distribution.
All right. Can I allow one more questions from the audience? Are the fat depots from the arms similar to the legs?
Oh, that's an interesting question. I think they're actually different. So evolutionary, perhaps we think of humans moving from quadrupedal to bipedal individuals, but we conducted a study that was published last year, a GWAS study, actually using DEXA features, where we for the first time actually found discrete gene variants regulating fat deposition in the arm, but not in the leg. There are very few studies looking at arm fat in relation to whole body metabolic profiles. So that question is largely unanswered. It might be a difficult question to answer because the leg fat and the gluteofemoral fat is a much, much larger depot. So it is likely to have a much larger impact on the whole body. But I think the questioner is asking a very interesting question.
Okay, right. Thank you, Prof. Frederick. To the audience, keep sending your questions to us, and we will address them during the final session later on. Okay, so we would like to welcome our next speaker from UKM, Malaysia, Associate Professor Dr. Zahara Abdul Manaf. She is a senior lecturer and a dietitian at the Dietetic Program, Faculty of Health Sciences, Universiti Kebangsaan Malaysia. She will enlighten us with the topic on the association between chronotype, dietary pattern, and obesity in adults. Please welcome Associate Professor Zahara. Prof, you are still muted.
Can you see my slide?
Yes, we can see your
Okay.
slides. Mm-hmm.
Thank you for a very nice introduction. It is an honor to contribute to this webinar together with a group of distinguished speakers. Before this, Dr. Carmen has presented the fundamental principle about the dietary management for the obesity, and other speakers discuss about the anthropometric body composition. So I'm going to discuss, share the evidence and share some of our findings from our studies about the association between chronotype, dietary patterns, and obesity, focusing on adult population. I'm sure all of us are very familiar with this phrase, you are what you eat. So we are focusing more on what we eat, the portion size, the calories. So today I'm going to share with you how the timing of meal is associated with our health, particularly the obesity. The timing of eating has become a public interest, and the researchers, after a group of researchers in a Salk Institute in California, published their research findings from their animal study, where they fed two groups of identical mice with ad libitum or time-restricted feeding with isocaloric high-fat diet for 8 hours per day, and they for 12 weeks. And what they found that the mice that were fed ad libitum had higher body adiposity, higher insulin resistance, and less active. So this study shows that the food given in the normal sleeping period can disturb the synchronization and impair the body function. Every organism has its own timekeeping system to anticipate and adapt to the day-night cycle, which is known as circadian rhythm. Circadian is a Latin phrase which means around a day or 24 hours. So it is a daily behavioral, physiological, and molecular changes with a cycle length approximately 24 hours, and ensuring all the body processes are optimized at the right time of the day. I'm sorry if you heard it's very, in my house it's very heavy rain. So maybe you can see the you can hear the noise. All right. Circadian clock play a crucial role in many biological processes, such as sleep wake-up cycle, hormone secretion, cardiovascular health, glucose homeostasis, and body temperature regulation. And the circadian rhythm of the body are controlled by the central clock located in suprachiasmatic nucleus, SCN, located in hypothalamus in our brain, and also by peripheral and cellular clock distributed throughout the body. The master clock in our brain is strongly entrained by lights. However, the peripheral and cellular clock also responsive to meals, social time, physical activity, sleep-wake cycle on top of master clock. Okay, so as night falls, the master clock initiates the production of melatonin, a hormone that promotes sleep and help us to stay asleep throughout the night. And during the daytime, light exposure causes the master clock to send signal that generate alertness and help us to keep awake and active. This is our modern lifestyle. This is here is KL before MCO. Before MCO. People, there's so many eating outlet 24/7. So I'm not sure how this situation can go back to the original. So we have 24-hour food premise, and with the gadget, artificial light, and mobile phone almost 24 hours, this can lead to a circadian misalignment. So Ford, can you hear me?
Yes, Prof. You done?
No,
We are still stuck at the modern lifestyle slide here.
Yeah, because I cannot press the page down. Because it's very heavy rain in my house.
Mm-hmm.
So
Possibly the interference of the internet connection.
yeah. So it's stuck now, but you can hear my voice, right?
Yes, we can hear you clearly. It's only the slides not moving.
All right.
Mm-hmm.
Maybe I stop sharing first.
All right, sure.
And then I share. Share.
Okay. You can see your slide.
Ah, right.
Yeah.
Okay. So circadian misalignment is a state where the environmental and behavioral cycles are out of synchrony with endogenous circadian system. So type of circadian disruption or misalignment can be divided into two: environmental misalignment, which occur when environmental factors such as light dark cycle is misaligned with circadian central circadian clock, for example, in jet lag and shift workers. Okay, and the behavioral misalignment occurs when the behavioral, which include feeding, fasting cycle, is misaligned with the circadian central circadian clock. So, for example, disorder of eating pattern, disrupted eating, disrupted sleep, and social jet lag. So disruption of circadian rhythm often reported to increased risk of metabolic syndrome, health problems such as hypertension, dyslipidemia, insulin resistant, abdominal obesity in our body, and increased fat storage. And disorder eating pattern is also can cause circadian misalignment. A circadian system reduce glucose tolerance in evening by decreasing beta cell function. This is in normal typical circadian rhythm. Okay. However, in circadian misalignment, reduce glucose tolerance by lowering insulin sensitivity, but not affecting the beta cell function. So shifting feeding time to rest phase altered the normal circadian of resting energy requirement and reduce insulin sensitivity. For example, if we eat heavy meals during midday, midnight, instead of the day, because at night we should sleep, rest, and without not taking heavy calories. So food intake during biological night is often reported increased risk of metabolic disease. Chronotype is chrono is related to time. Is a morningness, eveningness preference, indicates individual difference in activities, as well as timing of sleep and wakefulness in adaptation to the light and dark cycle 24-hour day. And early chronotype tend to perform better in morning, while late chronotype perform better in the evening. So the morning chronotype, we always refer them as early bird, and evening chronotype often known as night owl. And the studies that looking at the relationship between chronotype with BMI are quite constantly reported that evening type had significantly greater BMI compared to intermediate and morning type. And compared to participant in normal BMI, overweight and obese participant reported with delayed sleep and then shorter sleep on the free days, and they are more likely to be evening chronotype. And there is also studies reported that among men, evening chronotype was significantly associated with the BMI. So in our recent reviews that is looking at the association between chronotype and diabetes among adults, we found that eveningness was linked to the delayed mealtime, skipping breakfast, excessive calorie intake at night, and eveningness linked to lower vegetable and high intake of sweets, sugar, coffee, and alcohol compared to morningness. But there's no difference in total energy, fat, and carbohydrate intake between the chronotypes. So in one of our study, we looked at the chronotypes, adiposity rate, cardiometabolic risk rate, and dietary practice among overweight and obese non-shift workers. So almost 300 participants, age of around 40, and about 70% are women, and they are all overweight and obese, and 99% of them are Malays. That's why we need to modify monitor chronotype questionnaires because most of our participants practice split sleep because in the morning they wake up for the prayer and then they go back to sleep. So we cannot use the standard MCTQ questionnaire to estimate their chronotype. So what we found was in terms of the adiposity, there's no difference because all of them are overweight and obese, and we didn't find any difference in terms of the body composition, but all of the cardiometabolic risk factors are significantly different and higher in the evening chronotype compared to the morning chronotype. In terms of sleep duration between chronotype in both working days and free days, evening chronotype had significantly less duration compared to the morning type. And evening chronotype is also show social evidence of social jet lag, which is a proxy of circadian misalignment, behavioral misalignment compared to the chronotype. We didn't find any significant difference in eating total eating window between chronotype. Total eating window is the duration between the first meal of the day and the last meal of the day. And but we found that the morning chronotype had earlier time for first meal and last meal compared to the evening chronotype. So as you can see, the evening chronotype tend to take later breakfast and dinner. The rain is very heavy. I have to fight with the thunderstorm. All right. And there's no significant difference in total daily energy intake between chronotype. This is consistent with a few previous studies. However, there was a significant difference in distribution of energy according to the eating window between the chronotypes. The morning chronotype tend to consume more percentage of energy and carbohydrate in the early eating window, and the evening chronotype tend to consume more calories and carbohydrate and fat during the late eating windows. We conducted a weight reduction program among these participants, but we haven't completed the data analysis. But the initial data analysis shows that the the morning chronotype reduced significantly more weight compared to the morning chronotype. Although there's no significant difference in terms of the calorie intake. A study conducted in Boston, USA, and published recently, they found that they compare late eaters compared to early eaters. Late eaters, most of them are evening chronotype. So they found that late eaters are more likely to have higher baseline obesity and lower morning appetite, and then they have more weight loss barriers, and more likely to eat when they are stressed and overeat at night, night eating syndrome, and tend to eat while watching TV. And they also found that each hourly delay in midpoint of eating is associated with 50 gram less weight loss in the weight loss intervention. And 3-hour delay in midpoint of meal is resulting in delay, which result from the delayed breakfast and late dinner timing contribute to about 150 gram body weight less weight loss compared to the early eaters. So in conclusion, eveningness is linked to greater social jet lag, delay meal time, skip breakfast, higher calorie intake at night, and consume less healthy calories. Despite some of the study shows that the total calorie may not differ from the two chronotypes. And obese or overweight individuals with evening chronotypes are more likely to be late eaters, higher cardiometabolic risk rates, higher weight loss barriers, lower motivation for weight loss, and lower efficacy for weight loss in weight loss intervention. And the relationship between chronotype with BMI is more consistent in the follow-up studies as compared to the cross-sectional studies. So in terms of the significance of the chrononutrition research in clinical practice, chrononutrition-based approach in weight management could be recommended to improve the metabolic profiles, particularly among evening chronotypes. So these can be the adjunct strategies on top of the basic principles, dietary principle that was explained by Dr. Kamal just now. Maybe the chronotype concept can be the adjunct strategies which involve behavioral therapies by advancing dinner, restrict duration of eating window, and eating a greater share of total calorie intake earlier in the day, and also synchronizing a sleep-wake pattern with 24-hour circadian rhythm. I acknowledge my research team and the Ministry of Higher Education for a funder for our research project in this. Actually, this is a early findings that we share, which we just conducted recently. And with that, thank you. The rain is stopped now.
Thank you, Dr. Zahara. Very interesting to learn on how the differences in chronotype affecting the cardiometabolic risk factors. So here is a question for you posted on the Facebook. For those working in shifts, such as nurses, factory workers, what is your suggestion for them to maintain healthy diet and normal weight, since they are pretty much affected by this? They belongs to the different chronotypes from the common people.
They have a misalignment of these circadian rhythm. So the best thing to do is shorten the eating window. Instead of nibbling throughout the nights, shorten the eating window, and if possible, not to expose themselves to the a lot of light at night, you know, because this is a the circadian rhythm will give signal that it's 24-hour daylight for them. So and then I think the best advice based on the evidence is shorten the eating window. Yeah.
Right.
Yeah, not 24 hours.
Not 24 hours.
Yes, the evidence shows that between 8 to 12 hours eating window is the best.
Mm-hmm. Shorten the eating window.
Yeah.
All right.
Yeah.
So thank you.
12 hours. Yeah.
Mm-hmm. Thank you, Dr. Zahara. So more questions will be allowed for at the final session. All right. So moving on, we would like to welcome our next speaker, also from Malaysia, Professor Datuk Dr. A. Rahman A. Jamal. Professor Rahman is a professor of pediatric hematology, oncology and molecular biology at the Faculty of Medicine, UKM. He is the project leader of the Malaysian cohort and also an active member of the Asia Cohort Consortium and Public Population Project in Genomics Consortium of Biobanks. Professor Rahman will speak on the Malaysian cohort as a valuable resource for research in non-communicable diseases. So please welcome Professor Rahman.
Yeah, thank you, Xiao Fong, for the kind introduction. Welcome to all the panel speakers and the audience. Thank you to the organizing committee for inviting me to share our experience in setting up the Malaysian cohort, which has now become a valuable resource for research, especially for non-communicable diseases. Well, there are many cohort studies and biobanks that had been set up, and many of these emerged in the early 2000s. Of course, UK Biobank, Prof. Sarah Lewington has mentioned that. There is a Taiwan Biobank on our side of the globe, recruiting 250,000. The Singapore Chinese cohort study or Singapore Chinese health study has about 68,000 people. It started in the late 90s. About the same time as the Shanghai cohort study, looking at middle-aged individuals. All of this is from the Precision Medicine Initiative from USA, aiming to recruit 1 million individuals. On the Middle East side, we have the Qatar Biobank. They are aiming to collect about 60,000 individuals, and sequencing all the genomes of these individuals. And of course, last but not least, that I'm showing here is our own Malaysian cohort that has recruited more than 120,000 participants to date. Well, in the year 2009, the Time magazine listed biobanking or biobanks as ideas, one of the ideas, top 10 ideas that would change the world. And in 2019, fast forward 10 years later, the Forbes magazine published an article mentioning that biobanking is changing the world. As you all know, the relevance of having a large population-based studies, which will include collection of biospecimens or biobank, would provide the opportunity to do large-scale research in diagnostic and therapeutics, big data analytics. It will also allow biomarker discovery and translational research, allow us to identify risk factors and associate this with diseases. And of course, as we talk about precision medicine now, biobanking and large population-based study would be a strong enabler for precision medicine. Of course, at the end of this, all these results would transform healthcare. And now we have the artificial intelligence driving a lot of the processes for biobanks and also cohort studies. Let's just introduce Malaysia here for the benefit of our guests from UK. We have about 32 million population and about 6% are aged 65 years and above. A multi-ethnic nation. Our health indicators are not too bad. They are quite up there with the developed nations. We have a very strong and comprehensive subsidized healthcare, almost universal in nature. With just 25 US cents, we could get access to healthcare. But with this, we have the sort of, in a sense, the indicator to show that our health is not that good. Prof. Nolaila has mentioned about the NHMS survey, type 2 diabetes in adults now stands at 18.3% prevalence. If you look at the top principal causes of death in Malaysia, this is from the Department of Statistics, Malaysia 2020. There's the dominance of NCDs as the main causes of death. Only pneumonia come into the top 5. The rest are related to non-communicable diseases. So how did the Malaysian cohort started? So there is a political will here. You know, when our Prime Minister in 2004 met with a Nobel laureate, Professor Leland Hartwell from the Fred Hutchinson Cancer Research Institute. And Professor Hartwell told him about large-scale population studies that are being sort of set up across the world. And at that time, Fred Hutchinson Cancer Research Center was hosting the Asia Cohort Consortium. And immediately, our Prime Minister decided that Malaysia should have a cohort study. That's how it all began, and the rest is history. Yeah. So now we have become a member of the Asia Cohort Consortium and the newly formed International Health Cohort Consortium, of which the Biobank is also an established member there. It took 2 years of planning, many workshops involving many experts. We had 9 subcommittees. We had an advisor from Fred Hutch itself, Professor John Porter. And well, we had a good time looking at protocols from both UK Biobank and also this side, from the Korean Biobank, from the Singapore Chinese Health Study, and so on. So we sort of had the best of examples to follow. Of course, we need assistance, and this is taken about 10 years ago, where even I didn't have gray hair then, so all looking very young. So these are the people on the ground that helped to build up this cohort. Many have left for better jobs, and we have a high turnover of research assistants because the Malaysian cohort is a good place for them to get work experience and become a skilled worker. Okay, we strategize to target 100,000 participants in a purposive sampling mode, as well as a cluster sampling. We oversample the Indians because they are a minority. We have the urban and rural participants as well. These are the location of the recruitment centers. So imagine our teams going out to the rural and urban areas, setting up recruitment centers, taking measurements and taking blood specimens and sending them back to our central processing center. So we decided also to use the geographical information system mapping. If you see here, each dot represents a participant, and this is very useful as we move towards environmental-based studies. For example, the role of air pollution in relation to lung diseases, for example, and useful for studying gene-environment interaction as well. We can also actually layer the location of fast food restaurants on this map, and also the sort of people who have diabetes, obesity, or cancers, and see whether there are clusters or patterns in terms of the distribution of these diseases. We measure them and we follow again the gold standard of many other cohorts, especially the UK Biobank. We were lucky that UK Biobank started about the same time. We measured the height, weight, and so on, blood pressure, ECG, spirometry, body composition analysis, and the cardiovascular index that can measure both arterial stiffness and also the vascular age. Coming soon is DEXA scan, as what we're doing for this project. We have started measuring this in a few hundred participants, retinal scan and echocardiogram for a sub-cohort. Biospecimens, we follow directly the standards from the UK Biobank. Very thankful with all the protocols that UK Biobank has produced and published, especially in terms of optimization and so on. So we have our own transport vehicles to ensure biospecimens are sort of not affected in terms of quality. They were sent to our central processing center within 12 hours of being taken. So these are the list of the blood and urine tests, quite standard. But what we are aiming to do in our study, in our obesity and diabetes study, is measuring insulin level. Our biobank is the largest in Southeast Asia, and we also have DNA already archived at room temperature that makes it simple for us to access DNA for GWAS or genome-wide association studies. Our bioanalytical laboratory is accredited with the ISO 15189. This lab does all the renal profile, full blood counts, lipid profile, and so on. So we had a target of 122 participants a day, and in September 2012, we reached our target of 100,000. In fact, we surpassed that. And we actually achieved 106,527 participants. We published this, our baseline data, in the reputable journal, International Journal of Epidemiology. And this is the distribution of gender in our cohort. Jennifer has shown the ethnicity distribution. I will skip this. And this shows the age distribution. As you can see, most are in the 40s and 60s. We recruit those above 35, just like many other cohorts in the world. So this is the sort of summary table for the prevalence of diseases for the 106,000 participants as published. The prevalence hypertension, 46.5%. But what's interesting is how we pick up new cases. And also for type 2 diabetes, 16.6%, 5% new cases. They never knew they had diabetes. And impaired fasting glucose. And the interesting thing as well is that we have data on their follow-up 5 years after their baseline recruitment. Obesity, this is the WHO sort of index. If we use the Asian sort of criteria, this would go up to about 30%. And hypercholesterolemia, people are always amazed at how come Malaysians have high cholesterol levels, never seen in the world, but somehow we are not proud of this. So I believe this is due to the kind of food we take. They're very tasty, but of course they have a high cholesterol level. So there need to be a lot of awareness programs on this. But of course, this gives a lot of opportunity to look at disease variants and also intervention studies. So I just want to show the snapshot of some of the studies. We did our first GWAS for type 2 diabetes, and this has been published in 2015, leveraging on the differences in prevalence of type 2 diabetes amongst the ethnic groups. As you can see, Indians have the highest prevalence compared to Malays, and Chinese have the lowest prevalence. And we published this and showed the variance in 7 key loci for type 2 diabetes in the Malaysian population. 2 of these genes, as we know, also play a strong role in obesity: the FTO gene and the MC4R gene. Yeah. So these are some of the completed research projects. You can Google the titles and the Malaysian cohort, and you will get this publication for your reference. Association of Job Sectors with NCDs. We published the reference values for spirometry based on 100,000 spirometry data. We also published the rural-urban dengue seroprevalence data, the first community-based seroprevalence data in Malaysia, actually. We also published the genetic risk factors for diabetic nephropathy, cardiovascular risk estimation using lipid profile and Framingham risk scores. We have not published this, genetic of sudden cardiac death, but this has been completed. We have completed a project looking at multiple assay of 25 trace, multiplex assay of 25 trace elements for detecting colorectal cancer. We also have completed work on whole exome sequencing, looking at new variants for familial hypercholesterolemia, and last but not least, gene-environment interaction to predict chronic kidney disease. So some of the ongoing projects, the list is not exhaustive here. We have many teams working on many different projects. Adiposity Disease Risk, this is the project we're talking about here. I'm thankful for the funding from MRC and Malaysia. Precision Medicine for Diabetics, that's our work with University of Dundee, also an MRC funded grant. Non-coding RNAs in diabetic nephropathy. We are starting work or planning to work on non-alcoholic fatty liver. Cancer biomarker discovery is ongoing using the omic science approach. We are planning an intervention study on pre-diabetics. Again, another group will look at mental health and cognitive function, especially looking at early dementia. Biomarker-based nutritional studies, and also some gene environment studies. I just like in the last few slides to look at the mortality, and since recruitment, we have recorded 7,575 deaths from all causes, and 1,506 deaths due to cancers. So these are the top 10 of cancer deaths in our cohort. They're quite similar to the national data, and this again because we have pre-symptomatic or pre-diagnostic sample. These are really valuable sort of data and specimens to detect biomarkers for early cancers. And if you look at this particular graph, based on the cancer death, we can see that many of them sort of die in terms of timing from enrollment to death. And if you look at the sort of accepted nomenclature for discovering biomarkers, death that happen within 18 months of recruitment are the best kind of specimens to look at for early biomarkers if you use proteomics, metabolomics, or even epigenomics. So we have also included a value added colorectal cancer screening for our cohort and simultaneously collects tools for our cohort biobank. As you can see here, we have published this data. From about 2,000 participants screened, we pick up 7 cases of colorectal cancer and more than 50 neoplastic polyps, which sort of have given added value to our cohort. So challenges, our health events are mainly via self-reporting because our hospital have not many hospitals have electronic medical records, and it's hard to link medical records to validate events. 40% of deaths are not certified by the medical personnel. We are planning to do verbal autopsy, and our Oxford colleagues are helping us a lot with that. The follow-up rate is about, we are past 60%, but we are still trying to trace the 40%, mainly due to changes in terms of phone numbers and also address and so on. So that's this is my last slide. In summary, the Malaysian cohort is certainly a valuable resource for research in NCD, and I believe the partnership with the Oxford University and also with Dundee and also through the International Health Cohort Consortium will really add value in terms of future research. The participants are well characterized with data on key exposures. It's a multi-ethnic cohort. That's the beauty of our cohort and the potential of what we can do with this. Urban and rural participants, biospecimens with high quality in nature, and we believe the value will increase with time. We have set up special interest groups to look at many different diseases. And I believe with our cohort, we have something on the table when we discuss with international collaborators, especially to do collaboration and also comparative analysis with other cohorts and biobanks. And last but not least, participation in global consortiums, and that would put Malaysia on the global map of populations, large population-based cohort studies. I'd like to thank the funders. Originally, the cohort was funded by the Ministry of Science, Technology and Innovation. And over the past 5 years, this has been funded by the Ministry of Higher Education, of course, UKM, where this project is sparked, and Umbi, collaborators and co-researchers, including all the speakers from Oxford today. And of course, all the Malaysian cohort staff that have been with previously and also currently. I think that is all. Thank you so much for listening. Thank you.
Thank you, Prof. Rahman, for the wonderful sharing. Here is a question for you from the audience. What is your comment on the validity and quality of the archive specimens? How close does the archive specimen resemble those that were collected in real time for analysis in order to reflect the true association between non-communicable diseases and the risk factors? What is your take on that?
Well, there are many publications that have sort of validated the quality of archival specimens over the years. And even UK Biobank has published some of this. And for us, we have also done a sort of analysis of samples taken at baseline and 5 years along the line to look at whether the parameters that were measured, analyzed included, including lipids and certain metabolites, whether they remain stable. So mind you, these samples are kept at in liquid nitrogen, that's at minus 196 degrees centigrade. And we have working aliquots at minus 80 degrees. So there are certain metabolites and elements that are unstable at certain temperatures. But so far from our reading, almost all the metabolites and analytes, if you keep them in liquid nitrogen, they would be stable, in fact, for many decades in future to come. So not to worry about that. That's the reason why there are many biobanks that have been set up, and sort of analysis can be done in future, and then correlate with what happened during recruitment. Thank you, So Fong. Good question.
Thank you, Prof. As usual, more questions will be allowed at the final session. So, okay, so last but not the least, we would like to welcome our speaker also from Malaysia. She is Associate Professor Dr. Nur Azian Abdul Murad. Dr. Azian is a senior lecturer and the director of the UKM Medical Molecular Biology Institute. She is the Malaysian PI for the MRC UK Malaysia Joint Research Partnership on Non-Communicable Diseases. She will present to us DEXA body composition, a preliminary result from the UK-Malaysia Health Research Partnership. So let's welcome Associate Professor Nur Azian.
Assalamualaikum and hi everyone. I'm going to talk about our UK-Malaysia Health Research Partnership for Non-Communicable Diseases project on obesity. I will cover the methods to determine obesity, including the conventional anthropometric measurement and the gold standard methods. Then I will discuss the prevalence of obesity in our Malaysian cohort project, and finally to share with you some of our preliminary results. BMI and waist-hip ratio are two conventional anthropometric measurements used to determine obesity and overweight. BMI is the most commonly used, and it is defined as weight in kilogram divided by height in meter square. As discussed earlier, based on the WHO, BMI more than or equal to 35 is defined as overweight, whereas BMI more than or equal to 30 is defined as obesity. Another common instrument to determine obesity is by using waist-hip ratio. According to the National Institute of Health, a waist-hip ratio of greater than 1 indicates that you have high risk of developing heart disease compared to normal individual. And a healthy waist-hip ratio for women should be less than 0.8, whereas a healthy waist-hip ratio for men should be less than 0.9. BMI and waist-hip ratio does not account for body composition, for example, measurement of muscle, fat, and bone. Thus, it may not be accurate or ideal methods to classify obesity. And the gold standard methods to determine overweight and obesity are DEXA and magnetic resonance image, or known as MRI. DEXA is a type of medical imaging test, and it uses very low levels of X-ray to measure bone density as well as body composition. Bone density can be used to diagnose osteoporosis, and body composition will give in-depth analysis of fat tissue, muscle mass, and bone density. This table here shows the measurement that can be done by DEXA, including total body fat percentage, fat mass index, visceral adipose tissue, android to gynoid ratio, and so on. For example, in this android and gynoid ratio, it describes the storage of fat in the body. Android or apple shapes is a condition where you have most of the fat around the stomach and midsection of your body, whereas the gynoid or pear shape refers to having the fat stored around your hip. This is an example of the DEXA machine. It is an open design machine and the participants just need to lay down on the machine. It uses low X-ray energy and measures two composition within your body. First, the soft tissue, and the second is your bone. The results obtained from DEXA will be in percentage, mass, and images. And this table here shows an example of the body composition results. This figure here shows the sex-adjusted prevalence of obesity and diabetes by age and ethnicity in the Malaysian cohort project. And the prevalence of obesity and diabetes in Malay and Indian are almost the same and are less in Chinese. Obesity will increase the risk of cardiovascular diseases and insulin resistance. Abdominal obesity and metabolic syndrome will lead to formation of ectopic adipose tissue, vascular deregulation, and inflammation. Altogether, these factors could lead to insulin resistant, atherogenic formations, high blood pressure, and atherosclerosis. Now I will touch a bit on the molecular basis of obesity. Obesity is caused by complex interaction of genetic factors as well as the environmental factors, and both factors disrupt the energy homeostasis and influence the susceptibility to obesity and other NCDs such as diabetes and cardiovascular diseases. South Asians have high prevalence of type 2 diabetes, and body composition could increase the risk of type 2 diabetes in this population. There is high evidence of disproportionate prevalence of type 2 diabetes in Asian, and this could be due to differences in body composition. In this study, the authors combined two cohorts from the Masala and MESA cohort. A total of 2,615 participants involved in this study, and here are some of the demographic and clinical characteristics measured in this particular study. They showed that South Asians have high prevalence of glycemic impairment and type 2 diabetes compared to other ethnic groups. However, there was no strong association that could explain differences in the risk of type 2 diabetes could be due to body composition. In another study in Korean population, BMI between 23 to 24 could increase the risk of diabetes significantly, and BMI between 26 to 28 could significantly increase the risk of CVD. Now I will talk about our work together with University of Oxford and the PI is Prospera. In this study, we will perform DEXA scans on 6,000 participants in the Malaysian cohort study. Here are the research objectives of this project. First, we would like to understand the relevance of obesity as a cause of diabetes and cardiovascular diseases in Malaysia. Second, we would like to understand the ethnic differences in body composition in Malaysia. And third, we would like to compare the body composition measurement using the conventional method and compare it with DEXA. This project enables comparative studies between the Malaysian cohort and the UK. And finally, we would like to establish shared resources and strengthening research capacity in both countries. We use universal sampling for this study, and 6,000 participants will be recruited from Kuala Lumpur and Selangor, nearby the recruitment center. We will try to get an equal distribution of ethnic groups consist of Malay, Chinese, and Indian. Obesity is classified based on WHO criteria. Participation in this study is by voluntary basis, and altogether we have about 32,800 participants leave near KL and Selangor due for their second and third follow-up. As you can see here, the percentage of overweight and obesity in Malay and India are almost the same, and it is less prevalent in Chinese. The data here shows the obesity classification based on BMI, stratified by ethnicity and gender. There were significant differences between ethnic group and BMI, similarly to comparison between female in different ethnic groups and BMI. The data here shows the obesity classification based on waist-hip ratio, stratified by ethnicity and gender, and there were significant differences between female in different ethnic groups and waist-hip ratio, as you can see in this figure. This figure here shows the mean total fat mass, mean android and gynoid fat mass from the DALYs analysis. Altogether, comparison between Malay and Chinese and India showed statistically significant differences in all criteria. No significant differences were observed in mean total fat, mean android and gynoid fat in Malay compared to India. These results are still preliminary and it is difficult to make any conclusion at this time point. From this study, we hope that we could have the detailed measurement of body composition using both conventional method and DALY machine. DALY-derived assessment will be verified using MRI, and subsequently, we can compare our results with the UK Biobank. Finally, we wish to develop the ethnicity-specific anthropometric guideline for accurate disease risk estimation for Malaysians to reduce the risk of CVD and diabetes in future. My take-home message to all of you: first, monitor your numbers, look after your BMI and waist ratio, increase physical activities, quit smoking, and eat healthy food. Obesity, diabetes, and CVD are preventable. Stay healthy, and together we could reduce the risk of NCD in Malaysia. And thank you for your attention.
Thank you, Dr. Azian, for the sharing and kind introduction to the UK-Malaysia partnership. We look forward to more meaningful data from the project. So here is a question for you from the audience. You mentioned about genetic factors in obesity. Are there any differences in the genetic factors from different ethnicity? And what are the genes reported for obesity among Malaysians?
Thank you, Dr. Jing. It's a very good question. So basically, genetic predisposition to obesity is very much depending on ethnicity and population, similar like hypercholesterolemia and other diseases, maybe. So basically, the genetic factors for obesity, it can be divided into two categories: the monogenic form and polygenic form. So basically, monogenic form, one gene can cause the disease, whereas for polygenic, many diseases, you need to have like many variants in several genes to form the obesity. So in Malaysia, I think there's a few projects have been done to look into the genetic factors of obesity. However, there are still at a small scale. For example, they just look at single gene or several variants. I think one particular study they have published in 2020, they've looked at a few variants using Agena. Okay, so basically from that particular study, they found that BDNF variants in BDNF is associated with high risk of obesity. But yeah, the variants involved in obesity is very ethnic dependent. For example, the FTO variants, it is associated with increased risk of obesity in European, but not in Asian. Yeah, I hope that answer the question.
Right. Thank you, Dr. Azian. Okay, so we have come to the final session for this webinar. In this session, I will read out some questions posted by our audience for our speakers today. Some of the similar questions will be read out in a collective manner, yeah? So we have several questions here. Prof. Frederick has been very popular, it seems. Okay, the first question to Prof. Frederick. Interesting sharing on human fat depots. What is your comment regarding thrifty gene hypothesis on human fat depot, and does it evolve with the urbanization of diet and lifestyle changes?
It's a big and complicated question. I mean, the thrifty gene hypothesis was formulated by John Neal nearly 50 years ago, before the human genome was sequenced. So a very theoretical concept. I'm not sure we have data to actually support the thrift gene hypothesis today. Possibly there is data to support it, but I think there is a lot of things we cannot explain using conventional genetic tools that explains body composition, body fat distribution, energy expenditure, satiety signaling, those type of gene environment interactions that we are actually thinking of about the thrift gene hypothesis. I think there is adaptations. Animal models show this beautifully of non-genetic effects, epigenetic effects on obesity across generations. And I think this is the place where I think one would really want to go. And that is not coming back to the thrift gene hypothesis. It's adaptations to environments through non-genetic methods. Siok-Fong, you are muted.
Sorry for that. Thank you, Prof. Frederick. I hope that answered the question from the participant. Okay. There's another interesting question put up here. How can we confirm that our weight is purely due to our genetic makeup? This seems like the case of putting the blame of being obese to our genes. So perhaps the molecular biology scientists would like to answer this. Prof. Frederick or Datuk Rahman, or maybe Dr. Azian?
I'll pass to Frederick.
I'll give it a start,
Yeah.
and I hope someone else can give a more intelligent answer than I come up with. But if we look of what the discovery GWAS studies have come up with so far, we see for obesity a very large number of genes that each and one of them contribute a very small proportion. But the overall explanation of heritability of adiposity is still very small. So I think there is more work to be done here. The heritability studies might overestimate the actual heritability, but there is more to be found here that we cannot explain so far. And when it comes to fat distribution, the gap is even greater. So it's a real dilemma.
Mm-hmm.
Yes, if I can add.
Coming back to the question, should we blame the genes? I mean, I think that is a very simplistic way of looking at things. I think what we have heard today from so many speakers is that it is a much more complex association where urbanization, change in diet, play on our genes. And that is what we should look at rather than single out a gene is bad, it leads to obesity.
Very true. Dr. Rahman, would you like
I
to
just
add?
want to add, even GWAS studies have shown that if you have variants that give you a relative risk, a certain risk of developing obesity, lifestyle sort of changes, even diet, can actually lower that or modify that risk. So meaning like what Professor Frederick says, the genes are not the most significant driver. You could still modify this through good lifestyle changes and dietary modification. Yeah.
I completely support that. And I think it's a very important statement. It's a very, very important statement. And it goes much further than just looking at GWAS and the small gene variants. We have examples of monogenic disorders that we believe have full penetrance for fat distribution, the partial lipodystrophies. And they are also following this pattern in a very, very clear way. Physical exercise overcomes these metabolic problems that you have.
Mm-hmm. So no blaming game here.
No.
Maybe
Right.
I add on Xiao Fong. For example, the RS6265 in BDNF, where my camera. Okay, so that particular variant is actually associated with increased risk of obesity when you are smoking. But if you are not smoking, it will, reduce the risk of getting obese. Yeah, so that's because obesity is a complex disease. It involves interaction between environmental factors and genetic factors, so it can modify the risk.
True, very true. It's a multifactorial disease. Okay, so the next one is regarding the maternal obesity. Maternal obesity is also a recognized risk factor for the CVD development in the offspring. Can you comment on what is the relative contribution of this exposure to maternal obesity in utero to offspring CVD risk compared to conventional risk factors? The audience directed this question to Prof. Sarah. Prof. Sarah, would you like to comment on this maternal obesity?
I'm not an expert in this area at all, so I'm giving my opinion. But I do think that there are studies that were done sort of towards the beginning of the century. So this was a huge hypothesis towards the end of the 20th century. But then actually, if you look carefully at confounding, so it's the environmental factors of the maternal, what's led to the maternal obesity, and then those environmental factors also affect the offspring. So I think you have to be really very careful when you're looking at these in utero risks going forward into future generations. I'm not saying it's not there. I think there's a lot of work to be done, and I think our work on the adiposity have seen these very different associations in populations that are maybe undernourished or not overnourished as children, but then being well nourished, potentially overnourished as adults, seem very different relationships with, particularly with BMI. But certainly with adiposity. So I think there's a lot of interest in it, but I think you have to be very careful when you look at in utero risks on the offspring, that you look very carefully at other confounding factors of the sort of cardiovascular risk environment. If anyone else from the panel would like to add to that, or maybe have a different opinion.
Any of the panel would like to add to this maternal obesity? Okay.
I can just make one comment. I completely agree with Sarah on this. She's a very level-headed, good response. The problem we have is that the actual research question is nearly not tractable. We don't have access to the tissues where we want to look for these types of regulation. Everything points towards that the genetic background towards obesity is regulated by satiety in the brain. So if we would like to look at these relationships that are questionable, we would need to look at epigenetics in the brain. This is not possible.
True. Okay, so right. I think we do have several questions regarding the weight loss. The first one is, many people are trying to lose weight by practicing keto diet, which is high in fat and low in carbs, made popular by the celebrities. So we do know that high fat is associated with higher risk of CVD. Do you think it is a safe approach? What would be the best approach to lose extra weight and maintain a healthy one? So perhaps Dr. Carmen or Dr. Zahara would like to take this question.
Well, I think I've said in my presentation that whatever diet that you can maintain long term, it's going to work. So a keto diet, especially if it's very, very low carb or zero carb, as people like to say, it's almost impossible to maintain, because at some point you have to eat some vegetables to avoid constipation, for example, and vegetables have some degree of carbohydrate. So a zero carb diet does not exist. And how long can you maintain it? And then the question of if you have very low carb diets, then you've got high protein, which most likely comes from animal sources. And then animal foods are coming with the issue of the fat quality, which I haven't really discussed in my talk, but all the other panelists can discuss about the fat quality, how important that is for cardiovascular disease prevention. So in terms of losing weight, I think the evidence shows that anything you can maintain long-term is going to wait. In terms of preventing diabetes or cardiovascular disease, the quality of the fat, the quality of the carbohydrates are going to be important.
Mm-hmm. True. Prof. Zahara, would you like to add?
Yeah, I agree with Dr. Carmen. Based on a lot of reviews about the ketogenic diet, even the celebrities or the founders said you can eat as much as you can, but if you follow the ketogenic regime, actually you end up eating less calories because of the limited food choices. So the participant lose weight because of the low calorie diet. And there's no long-term studies in terms of the safety of this ketogenic diet. And there's an issue about the adherence to the diet for the long term. So to kickstart of weight loss, it is okay, but in maintenance, it is not about, it leads to the weight loss is due to the low calorie diet basically. But there's another issue to think about, the adherence for long term, the safety of the long term consumption of ketogenic diet is still need to be considered. Thank you.
Thank you, Prof, for the sharing. It seems all right. Yeah, it seems that the key for this is always consistency. Whatever diet you're on, just be consistent, and calorie restriction. Okay, the next one, I guess it's Prof. Laila. It's directed to Prof. Laila. Is Prof. Laila still okay?
We'll see her here.
Yeah.
She's left.
Yeah, perhaps Prof. Laila already left. There's a questions on diet and exercise. The participants wrote here, diet and exercise are not working for me. What else can I do to treat obesity? So I guess we open this for our panels. Anyone would like to share your opinion on this? If diet and exercise are not working, what else can I do?
I'm not sure I want to answer that, because it really depends on what's been tried before. I mean, I have discussed the evidence on non-surgical interventions, behavioral interventions. So of course, we've got all of these other portfolio of surgical interventions, which I'm not an expert on and I can't really comment on, which do produce weight loss, but they also have weight regain. So we have to also keep that in mind. And they have other issues in the long term. So yeah, so it's impossible to know what could work next for that person without knowing what he's tried before.
The best is go to the doctor. Go to the doctor and ask for professional advice.
Yeah, well, bariatric surgeons would promote bariatric surgery, not only because it works to sort of have obesity and can reverse type 2 or sort of early type 2 diabetes, but for our hospital, it also helps in income generation. I shouldn't mention it, but that's the most popular surgery that making our hospital, especially our private wing, earns more income. Just on the lighter side.
All right. Thank you, Prof. Rahman, for
Add
that.
one thing to that. I think, wasn't there an article published last month or earlier this year on a new intervention for weight loss that seemed to be effective? It was published in one of the American journals. I'm sorry, I can't remember the name of it. I don't know if anyone else saw that.
Was it on like intermittent diets or the 5-2?
No, it wasn't a lifestyle. It was pharmaceutical.
Oh, pharmaceutical. Okay.
Yeah, no, Sarah, what you allude to the semaglutide study in New England Journal of Medicine, which of course nearly a first pharmacological study that doesn't have severe side effects that are dangerous and provides weight loss. I mean, it has side effects that are unpleasant for the participants, but they seem to cope with that in the study because the weight loss was so profound. I'd like to go back to the question about exercise, because there is abundant evidence that what you achieve with exercise is energy deficit. And we are evolutionarily engineered as human beings to preserve our energy. So exercise will trigger overeating. So we are not losing weight with exercise. And that has been shown over and over again. You achieve other things that are very beneficial. You probably exchange a little bit of fat mass for lean mass. You get a better metabolic profile. You get all those type of benefits. But exercise is per se is not a useful way to lose weight if that is your aim.
Yeah.
True, very true, Prof. Okay, the next one, I guess it's regarding the chrononutrition. Recently, chrononutrition had been in spotlight. What would be the future for chrononutrition? And could we see the implementation of chrononutrition as a treatment for pre-diabetic or obese patients? So perhaps, Prof. Zahara, it's very obvious. The question is for you.
I won't say that chrononutrition is the sole treatment modalities for obesity and diabetes. It should be used as adjunct, added to the basic principle of dietary management that we always prescribe to the patient. So, but it basically what the chrononutrition research found that is actually in line with the healthy diet that we always, but it just that we focus more on the calorie balance and the portion size, maybe less on the timing, and relate with the circadian rhythm. So I think there's a study look at the effect of isocaloric diet in the weight loss program, and those who consume higher calorie in the late time of the day, in the evening, reduce less body weight and higher glycemic profile compared to those who consume greater calories in early day. So I think is this not only reduce go for calorie deficit, but synchronize the calorie consumption throughout the day will be the added value to the outcome of the treatment.
All right. Thank you, Prof. Zahara, for the answer. I hope it does answer the participant on this. Okay, in the interest of time, I think I'll allow the one last question. This is to Dr. Carmen. What is your opinion on snake diet approach introduced by Cole Robinson? Is it advisable or applicable to everyone who struggle to reduce their weight?
Sorry, what diet approach again?
The snake diet approach. Snake diet.
Oh, I haven't heard of that.
Yeah.
The snake diet.
Snake, yes, snake.
Oh,
snake.
gosh. I mean, there are. I think I can guess what the snake diet is about. And it could be like there are thousands of snake diets out there. Haven't heard of that one exactly, but some of them are just like some short-term trendy things that they're not based on evidence. I wouldn't follow anything that's not based on evidence. So, anyone heard of the snake diet out there?
Snake
No?
diet. Yeah.
I've Googled it, and the first thing that come up on Google is that
I
it
just
has to do
Googled.
with intermediate fasting, possibly.
Yes.
Prolonged fasting, yeah.
Oh, and what is the snake? Where is the snake come into play?
The prolonged fasting diet. Yeah.
Is it like feeding your food to the snake instead of yourself?
I don't know, maybe you drink some snake juice. The second hit had something about drinking snake juice.
Oh, goodness.
No, I think because like a snake, it takes one large meal, then it goes to sleep for a long time.
Okay.
So that's a snake diet. They eat a
Yeah.
lot, they binge, and then they rest they fast for a long time. So that's what it said, and I Googled it as well.
It is imitate eating pattern of wild animals.
Yeah.
Mhm.
It's
Did
like lion and all that. And bears, they fast for long and then they feast.
Feast on it. Yeah.
So there is a lot of trendy things about starving or fasting diets. I think we need more evidence on them in general. As I said previously, if your breakfast is going to be high sugar, high junk, it's better that you fast and skip that breakfast. But there are different approaches that are coming up every day about how you can fast half of the day and then eat all your meals in a specific number of hours a day. I don't think we have enough evidence about them. But from the physiological point of view, I'm not an expert, but maybe Frederick knows what is the benefit physiologically of fasting a few hours at the beginning of the day or later in the day on metabolic.
Maybe I could just add something to that. I think it's all about calorie restriction, isn't it? That if you're not eating for half the day, you're eating less. And those 5 and 2 diets showed that, yes, people eat more the day after they fasted, but not twice as much. And so I think it comes down to the point you were making originally. The main point is calorie reduction leads to weight loss, but whether doing that in these ways is good for you other than weight loss, I think is very controversial.
Yeah.
The starvation type of diet. Okay, before we end this Q&A session, any last word or take-home message from each of the speaker? Let's start with Dr. Azian.
It's in my slide. So basically, just look after your BMI because DEXA or MRI is quite expensive. So just look into BMI and waist ratio. Get an ideal body weight. Yeah, normal. Just make sure it's normal.
Okay, Prof. Frederick.
Well, I'm sorry, Asian, I will contradict you. I'll be very controversial by saying there are better ways to measure the adiposity risk than BMI. For example, really considering fat distribution. And I think we need to use those type of simple tools to actually stratify patients for interventions. Just using BMI is too simplistic.
Okay. And on DEXA. If promote DEXA,
no, for a population, waist and hip would be fine.
As long as you control the weight, isn't it, and body fat. Okay. Any take-home message from Prof. Sarah?
Well, I think just reiterating that, I think it's about getting large studies, large population-based studies, that if we're going to really understand fat distribution and the differences, I think those data from Chennai show quite clearly, and your data from the Malaysia cohort, that BMI actually is a crude measure of excess adiposity that maybe works in well-fed fat Europeans and North Americans, but it's really not looking like a good measure of excess fat in the Asian populations.
Agree, Prof. What about Datuk Rahman? Any take-home message from you?
Yeah, I just, it's more programmatic in nature. I believe that this is the kind of collaboration that, you know, we looking forward to, comparing across international cohorts and looking at the differences between ethnicity. And what we have in the Malaysian cohort is really of value because we can look at 3 large ethnic groups simultaneously as we compare with other cohorts that may be equally diverse or maybe they're more homogeneous. So we look forward to more of this kind of collaboration in future. Thank you.
Thank you, Prof. Dr. Karman?
So I think my take-home message is that obesity is a very complex problem. And that's probably the reason why we haven't found solutions yet, because we can't just look for simple solutions for such a complicated and complex problem. And also to remind people that individuals need to be motivated and empowered to be able to lose weight or keep their weight, you know, within their normal healthy limits. But also the environment needs to help us to reach that target, and the environments at the moment are really not helping anybody. So I think we need a lot more from politicians, from scientists, and from the public in general to help us change the environment where we live, to help us all live healthier.
Very nicely said. Prof. Zahara?
Just a short message. Unfortunately, there's no shortcut so far.
No shortcut. Okay. Last but not least, Dr. Jennifer?
I don't have anything to add to what other people said, but it's been great to be here and to see all the research that's coming out across the globe, and it makes for a very exciting future. And hopefully we can answer some of these questions we don't currently know, and start being able to intervene and help prevent some of these actually very preventable diseases.
Okay, so right, for the participants, please feel free to approach any of our speakers offline via emails if you do have any questions in mind. We still receiving questions coming in, but because in the interest of time, we couldn't address all at the moment. So with this, I would like to thank all of our speakers today from Malaysia and also from the UK. And also thank you very much to our audience for staying with us throughout this webinar. And we wish you a great day ahead. Enjoy your evening and enjoy your morning. Right. Thank you.
Thank you. Thank you so much.
Thank you to everyone.
Bye-bye.
Bye-bye.
Thank you, Prof. Sarah. Thank you, Prof. Adri. Jennifer.
Yeah. Thank you. Bye.
Bye.
Thank you, Prof.